Okay, welcome to topic 2 for our first quarter in Biology 3 for this school year. So this is entitled the Morphological and Molecular Data from Phylogeny. So this will be a very short discussion because this topic is very practical. Okay, to start, let us review phylogenetics. When we say phylogenetics, this is a study or systematic study of reconstructing the past evolutionary history of extant species. When we say extant species, these are species that are living or we can observe nowadays, okay, based on present day data. So there are two types of data that we can use in phylogenetics. We have the morphological and molecular data. Another review about character. When we say character, it's a well-defined feature in a taxonomic unit that can assume one out of two or more mutually exclusive character states. For example, color, white or black. So if it is black, it cannot be white. If it's white, it cannot be black. Okay? So there are two types of characters. We have the homologous character and analogous characters. When we say homologous trait of an organism or a taxonomic unit, this is shared because of, or it is similar because of ancestry. But when we say analogous, this is similar features or characters because of convergent evolution or homoplasy. So when we say homoplasy or convergent evolution, they live in a similar environment. That's why they acquire a similar trait. Okay, and in phylogenetics, we are interested in homologies or homologous characters. Okay, so again, when we say homologous versus analogous, no, when we say homologous characters, it doesn't mean that homologous characters are of same function. It can serve different function, but they can be homologous. Okay, for analogous structures, because they are in the same environment, most of the analogous structures are of the same function. Okay, other term for analogous structures are homoplasy. Don't be confused homoplasy from homologous. They are opposite. Homologous and homoplasy are opposite terms. Okay, the synonymous to homoplasies are analogous structures. Okay, now how to distinguish homology from analogy? So when we say homologous characters, it can be distinguished from the analogous characters based on its complexity, and because the homologous characters is a character derived from ancestry, no, we can compare them using fossil evidences and also degree of complexity, because the more complex the structure is, the less likely it is analogous structure. For example, when we examine the skull of human and other primates, the groove or the break in the skulls, no, in the development of the skull, yung parang mga cracks noon, no? So the cracks are homologous to each other. It's almost similar to each other. So it is unlikely that the cracks is because of convergent evolution. Okay, it's more likely because of ancestry. Okay, and the more elements that are similar in two complex structure, the more likely it is that they are homologous. Okay, another example. Okay, for another example is this sugar glider that can be found in Australia and the flying squirrel that can be found in North America. Okay, so as you can see, the two organisms are almost identical. No? So you might think that these are related to each other. But when you examine the complexity of sugar glider and the flying squirrel in North America, the flying squirrel in North America is a mammal, okay? And this one is a marsupial. So yun pa lang, no, complexity pa lang ng reproduction ng sugar glider from the flying squirrel will tell you that the similarity of the features of these two organisms is due to analogous or convergent evolution because of the similar environment that they are in. Okay? All right. Homologies and analogies can also be found in molecular data. So in molecular data, the character is the sequences. Okay, and the character state is A, T, C, G. So pwede siya maging A, T, or C, or G. Okay, and it's very important also to distinguish a homologous sequence from the analogous sequence. For example, no, there are two examples of sequences here. So 1, 2 sequences and 1, 2 sequences. We are comparing them. So again, no, i-review lang natin yung sa sinabi natin sa homologies and analogies kanina. The more elements that are similar into complex structure, the more likely it is that they are homologous. So as you can see here, the more similarities they have, the more evident na ito ay homologous. So from these two, we can say that similarity between 1, 2, 3, 4, 5, 5 sequences here in this 2 DNA is due to analogy. And here, you can say that this is homology. Pero isipin niyo bakit pinag-pair ito tsaka yun, tapos yun tsaka yun ay merong nasa gitna. Remember the mutation in DNA, okay? It can have deletion, insertion, or substitution. Okay? So yung pagkakaiba nito dito, yung sa kanina natin, no? Ito, it could be attributed to a deletion and then an insertion. Kaya kailangan silang meron tayong tinatawag na alignment. So we use softwares to align sequences to grade yung similarity nila from each other. Alright? So yung mga software na yon ay gumagamit ng mathematical tools to identify molecular homoplasys or coincidences. Right? Alright, so para mas masigurado tayo na we are looking at homologous sequences, we are using special sequences in phylogeny. So we have universal sequence, tinatawag natin ribosomal RNA. Now, ano nung special dito sa mga sequences na to? These sequences can be found or are encoding for a very important component in the cell or in the organism. So konting change in these sequences could be detrimental to the organism. So the organism cannot afford to make many changes in these sequences. So pag nagkaroon ng changes in these sequences, we can say that this is due to ancestry. Okay, na napapasa siya through time. Okay, for universal or organism can use ribosomal RNA. For animals, kasi mahirap i-compare lahat-lahat. So if you want to delineate in the animals only, we have the cytochrome oxidase 1 and cytochrome oxidase B gene. Okay, for plants, we have what we call our RBCL gene and the MATK gene. It's in your learning guide. And for fungi, I think this is not in your learning guide. This is the ITS ng intergenic spacer region. So these sequences are being used in the DNA barcoding. So we have what we call DNA barcoding to identify easily the organisms. Okay, so like I said, these are highly conserved regions. Now, in homologies of sequences, bakit ba nagkakaroon ng homology sa mga sequences? So there are 3 types of homologous sequences. We have the homology due to orthology, and then paralogy, and the synology. When we say orthology, these are similarities due to speciation. So there had been change in the original sequence, in the ancestral sequence. And then, nagkaroon ng divergence. So nagkaroon siya ng unique sequence dito, which constitute to another species. Okay? Another type of homology is what we call the paralogy. When we say paralogy, it's due to duplication. Because of, diba, we came from a very simple organisms. So, para magkaroon ng complexity, nagdo-double yung gene. No, this doubling of gene is what we call paralogy. Okay? Another type of homology is what we call synology, which is due to horizontal gene transfer. When we say horizontal gene transfer, hindi sila magkamag-anak, but it acquire the genetic material of a far or a distant organism. For example, the acquisition of the chloroplast from a primitive cyanobacteria, and also the acquisition of mitochondria from a primitive bacteria. Okay, so nagkaroon siya ng merging of the DNA from the host DNA. So, yun. So, naghalo ng kanilang gene. So, that's what we call synology, or due to horizontal gene transfer. Kasi when we say vertical gene transfer is parent to children. So, when we say horizontal gene transfer, hindi siya parent to children. Right? Next topic is about construction of cladogram based on morphological data. So it's very easy. I think you have done this in your IS, but we will just review. Okay? So in your learning guide, we have this. Okay? So hindi ko na ata papasagutan yan sa inyo. So we have this table. So you were asked to identify different characters of these organisms. So forelimbs, skin with feathers, skin with scales, egg with shells, bony skeleton, amniotic egg, vertebrae, and hair. So when we fill out this table, we can have this table. So we have the forelimbs from monkey, crocodile, rabbit, frog, and bird. Feathers for the bird, and so on and so forth. Okay? Now, from this table, we can make a cladogram. So, making a cladogram, alin ang pinaka-common sa lahat? Of course, this is the presence of the vertebral column. Okay? Next, na common sa lahat is the bony skeleton. Okay? The bony skeleton. Lahat ay may bony skeleton except for one. So, we will separate the shark from the rest. So, naka-separate na si shark. Okay. Next, the presence of forelimbs. Sino ang na-separate dito sa forelimbs? So, of course, the catfish and the shark. Si catfish and the shark. So, yun na, si catfish and the shark. Next natin is the amniotic egg. Sino ang na-separate natin sa amniotic egg? Okay? So, syempre, ang na-separate natin dito ay si frog. Okay? Si frog na na-separate natin. And then, we have the presence of hair. So, presence of hair natin. Sino nang may presence of hair? The rest ay walang presence of hair. And then, we have the egg with shell. So, pwede natin silang i-separate sa merong hair tsaka nung egg with shell. So, nung merong hair, we have monkey and rabbit. And then, egg with shell, we have two organisms. One with scale, the crocodile, and the other one with feather. So, that is the bird. Okay, so that's very easy. Okay. Next is the construction of cladogram based on molecular data. Okay, it's a little complicated in the learning guide, or if you encountered the cladogram based on molecular data for the first time, it's a little bit complicated. But actually, hindi naman siya ganun ka-komplikado. Basta isa-isahin nyo lang yung steps. Okay. Why we use molecular data than the morphological data? So, alam natin, sigurado tayo, DNA is inherited. So, when we say inherited, it's ancestry. And we can easily and quickly, inexpensively, extract and sequence genetic material. And sequences are highly specific and are often information rich. Okay. Now, when we do molecular cladistics, meron tayong simple steps. Number one is experimental design. Ano ba nung gusto mong makita? Animal ba yan? Plant ba yan? So, nakita natin kanina, no, there are different types of special sequences for different kind of organisms. Kung halo-halo ng mga organism, of course, you will have to use the rRNA. But if you're just going to use animal samples, of course, the animal primer lang ang gagamitin mo. Okay? Next, collection of data. It's either you will extract the DNA yourself, no? You will collect samples and then extract the DNA. Meron naman tayong mga kits in DNA extraction. And then sequencing. Or we can just get existing sequences from the GenBank. Okay? And then after this, after the sequencing, we have sequence assembly and alignment. So, kailangan yung i-assemble kasi minsan hindi sila dugtong-dugtong from the DNA sequencing. So, kailangan siyang i-assemble and kailangan nyo silang i-align. And then, after that, magkakaroon ka na ng phylogenetic inference. There are two types of phylogenetic inference, the character-based and the distance-based. When we say character-based, these are methods that are comparing all sequences simultaneously, considering one character or site at a time. There are two types of tree based on character. We have the maximum parsimony and the maximum likelihood. When we say maximum parsimony, again, these are evolutionary tree that says that the minimum number of steps is the most parsimonious tree. Okay? The next character-based tree is what we call the maximum likelihood tree. The maximum likelihood tree tries to find the most probable evolutionary model in creating the phylogenetic tree. There are different types of models. And the second type, non-character-based, and then the second is the distance-based method. When we say distance-based method, we are comparing two sequences at a time. So we have two types. We have the neighbor joining, which provide an unrooted tree, and the UPGMA that provides a rooted tree. In our learning guide, we are to construct a cladogram using UPGMA or the rooted tree. Alright? So, in constructing a cladogram, so run through lang natin nung nasa learning guide niyo. No? So, you have here sequences, so you have to align the sequences so you can compare the different kinds of organism or the sequences of different kind of organisms. So you have to grade them one by one. For example, A to B, A to C, A to D, A to E, and then A to F. And then you have to identify the differences. So for example, here, the differences between A to B is 9 differences. So we have to put 9 here. And then A to C, 2. A to D, we have 4, so 4 there, and then A to E we have 9, and then A to F we have 11 differences. Okay, and then the next B to C, B to D, B to E, and B to F. So we have 9, 6, 2, 1, to 11. 9, 6 to 11. And next is we have to compare the C to D, C to E. Okay, D to E, D to F, and then E to F. So we have here, we have 5, 9, and 11. We have 5, 9, and 11, and then we have 6 and 10, 6 and 10, and then 10 there. Okay? Now, from this table, we have to identify which pairs have the least number of differences. So we identify that C and A and E and B has the least number of differences. So meaning they are closely related to each other. So meaning they are the first clusters. Okay, and then we have to treat these clusters as one group, and then rework the matrix. So this 9 here came from the A to B. So we have it A to B, and then A to E. So we have A to B, and then A to E. And then C to B, and then C to E. C to B, and C to E. So A to B is 9. A to E is 9. C to B, C to B. C to B. Okay. C to B. Yeah, C to B here is another 9. And then C to E is another 9. So 9 plus 9 plus 9 plus 9 over 4. So average, it's the average. So it means it's 9. So ayun, 9 siya. Alright? Okay. Okay, same thing with this one. Same thing with the other, this one, the 4.5. Where did we get the 4.5? 4.5 is from A to D, and then C to D, and then the average. So that's 4 and 5. A to D, and then C to D. C to D is 5. So 4 plus 5 divided by 2 is 4.5. Okay, so that is 4.5, and so on and so forth. Okay, so you have to rework the matrix. Okay, now from this matrix, we have to identify now which has the least number of differences. So we have the least number of differences, which is the D, which is the A to C, A and C to D. So we have here the A to C and D. Okay, so we have to rework again, no? And then we have to rework, and then we have to here. So this is A to C. So another. So we have the A to C. So we have the A to C, then A to B, A to E, and then B to B, and then D to E. So that's 7.5. Okay. So from that, from that table, we now have another table that, and we can identify which has the least number of differences. So that's A, C, D, and B, E. So we have the A, C, D, and B, E. So the least number of differences, or the most number of differences is the F. So we can put the F in the outgroup. So we have now a rooted phylogenetic tree. Right? The last is about inferring evolutionary history in the relationship of organisms. It's very practical in just how to infer or how to obtain information from a phylogenetic tree. Okay? So phylogenetic tree as a hypothesis. So remember, phylogenetic trees are hypotheses. So we can have the phylogenetic bracketing, which is a method of inferring on likelihood of unknown traits. So these unknown traits are traits that do not fossilize well. For example, the feathers, the hair, like that. So you can infer from the nearest extant relatives. For example, in this phylogenetic tree, these are already fossilized organisms. So from here, we can see that the birds, which is our extant organisms, are closely related to this dinosaur. No? So we can assume that these dinosaurs have feathers too. Parang ganon. Okay, instead of scales. Right? So that's it. So that's our lesson for the topic